Okay, everyone, this is the video lecture for Chapter 16. This is probably going to be the last chapter we'll do for the course, and this video lecture will be divided into at least two videos, I imagine, for this one. One thing about this video, or this chapter rather, in the PowerPoint that I normally show in class, there are two videos that actually show as part of this. One of them on what's called the Hershey-Chase experiment, and a BioFlix animation toward the end on DNA replication. For some reason, neither one of those is working in my iPad, despite various things I've tried to fix this. So those videos are on Blackboard, so you can go there and you can watch them. There are also a lot of other videos, which I'm not going to show, or normally don't, but they are there as sort of ancillary material. But importantly, the two videos that I would have shown you in this video, or in this lecture rather, are up on Blackboard, so you can go watch them. Okay. All right. So chapter 16 basically is about DNA. It's about the structure of DNA, the history of research into DNA, and also in the second half of the chapter, importantly, about how DNA replicates or copies itself. So in the first half, really, we'll be discussing the history of research into DNA. And this is very important because of this idea that I've hit on so often this semester of structure and function. If you want to understand how something works or why something does what it does, generally speaking, you need to understand its structure. And that's certainly true with DNA. And so much of the early research into DNA was involved with deducing or figuring out the structure of the molecule, because then that would hopefully lead to an understanding of how the molecule actually does what it does, which is basically code for hereditary information. All right. So we're going to start out talking about Watson and Crick first. So in 1953, two researchers, James Watson and Francis Crick, introduced a really nice model, a double helical model for the structure of DNA. All right, so DNA is deoxyribonucleic acid. Let me just write that down real quick. So deoxyribonucleic acid. All right, that is DNA. Okay. Today we know that hereditary information is encoded in DNA and reproduced in all the cells of our body. So you can think of this DNA program as sort of directing the development of all sorts of things, biochemical, anatomical, physiological, and we're learning more and more nowadays, even to a certain extent, behavioral traits. But again, understanding how DNA does this, in order to understand how DNA does this, we have to understand the structure of the molecule. And that's primarily what Watson and Crick's work back in the early 1950s was all about. All right. So here you see a picture of Watson and Crick. This is Watson here on the left. He's still around. This is Crick on the right. He passed away, I want to say, about 15 or 20 years ago. But this is them back in 1953 in their lab. And you can see that they sort of built this Tinker Toy-like model of the DNA molecule. Okay, it's a little bit hard to tell there, but this is more of a modern conception of what the DNA molecule might look like if you could actually see it. It's called a double helix. Helix in the sense that it's shaped like a spiral, and double in the sense that there are two strands, one here and one here, that run parallel to each other as that molecule spirals up. Think of the DNA molecule as sort of shaped like a spiral staircase, all right? It's a good way to think of it, or at least imagine what it might look like in your mind. Okay, so Watson and Crick today are famous because they are the ones who officially worked out the structure of that molecule. And what I mean by officially is that they were the first to publish a paper laying out the details of the structure of the molecule. So they sort of get credit for that. This is a very simple one-page paper, which is kind of unique as far as scientific papers go. More often than not, they're 10, 15, 20 pages in length. This one was about 1 page long, which is kind of ironic because it was so important in terms of its ramifications, right? This was detailing the structure of a molecule, DNA, that once we understood the structure of that molecule, it opened up the entire DNA engineering revolution that affects us today, whether we realize it or not. Genetic engineering, biotechnology, all of that resulted directly from Watson and Crick's work. This is important because whether you realize it or not, on an average day, about half of the food you consume contains some sort of genetically modified component to it. Many of the new drugs that we get from the pharmacy, from our healthcare professionals, antibiotics, things like that, are produced using recombinant DNA technology, taking DNA from other sources, other species, and recombining it to produce something new. And all of that is a result of our understanding of DNA, which goes straight back to Watson and Crick's work. All right. Okay, so we'll get back to Watson and Crick in a little bit. Let's go back a little bit further prior to Watson and Crick, though. So early in the 20th century, the first half of the 20th century, the major sort of issue to molecular biologists, the major challenge to biologists was identification of the molecules of inheritance. What molecules are responsible for transmitting information from one generation to the next? Today we know it's DNA, but in the early 20th century, that wasn't known. There were two primary candidates. DNA was one of those candidates, but also protein was one of the candidates as well. And that makes perfect sense because, as I've said a lot of times already this semester, protein does everything in our cells. It's the workhorse of the cells. So why wouldn't protein, or why couldn't protein act as the genetic material? All right. The other candidate, of course, was DNA. So you sort of had two camps of biologists. One camp or group who thought protein was the genetic material, another group that thought DNA was the genetic material. And a lot of molecular research going on in the early 20th century, from World War I to World War II, so on, during that time frame was involved or focused on trying to figure out which of these molecules was the hereditary material. Okay. Now, there was a biologist named T.H. Morgan who had a lab, and his research group, was the first to actually show that genes are located on chromosomes. So people, in the early 20th century obviously knew that chromosomes were structures that appeared during the cell cycle, the mitotic phase of the cell cycle, and they moved around, as we've talked about in mitosis chapter, and were distributed to daughter cells. That made chromosomes a good candidate for carrying genetic information. Well, Morgan's group was the first to sort of prove that heritable units, what we today call genes, are actually located on those chromosomes. But the problem was that chromosomes are made of both DNA and protein. Chromatin is a complex of DNA and protein. So just because Morgan's group found that genes are located on chromosomes, that didn't answer that question, what is the hereditary material? Is it DNA or is it protein? Because chromosomes are made of both. All right. The role of DNA in heredity was first figured out or discovered by studying bacteria and the viruses that infect them. All right, and this is going to get us into talking about the Hershey-Chase experiment. So, evidence for DNA as the genetic material came from studies of viruses that infect bacteria. Viruses that infect bacteria are technically referred to as bacteriophages. So this term phage, that means virus. And specifically, a bacteriophage is a virus that specializes on infecting bacteria. Okay, so what is a virus? Well, a virus is really not even thought of as being a living thing. I mean, there's some biologists who think they're alive. There are others that don't. I personally don't think that they're alive. And the reason for that is because they don't do all the things that living things do. But they are this weird sort of group of pseudo-organisms, if you want to call them that. For one thing, they're not cells. They don't have a cell membrane. All they are basically is a protein shell with genetic material, either DNA or RNA, packed inside of that shell. That's it. They don't have organelles. They don't have a nucleus. They don't have membranes. You can see why some of us think that they're really not alive. But the important point is what I just mentioned. Viruses are made of DNA and protein. Some of them have RNA rather than DNA, but they're made of nucleic acid and protein. All right. And remember, we need to figure out, is the hereditary material DNA or is it protein? All right. So let's look at a virus. So this is a virus right here. This little guy sort of color-coded in green. That's a virus. So we have an outer shell made up entirely of protein, and this one has DNA packed into what's called the phage head. All right. This is a bacterial cell. That's the cell wall of the bacterium, and this guy has landed on that like a little moon lander, so to speak, and it's injecting DNA into that bacterial cell. This is what viruses do. This is how they take over cells. What's going to happen is once that genetic material from the virus gets incorporated into the bacterial DNA or genome, it will take over. It will reprogram that bacterium to become a virus factory. And so that bacterium will stop doing what it normally does, its own functions, and it will start assembling viral components. It'll put together maybe 150 or 200 new baby viruses inside of that bacterial cell. Then the cell lyses or tears open, and all those new viruses go out in the environment, and then they can infect more cells. So you can imagine how quickly a virus can spread through the body in this way. All right, well, it was generally known during Watson and Crick's research days, when they were doing their research, how viruses do this. But the problem was that viruses didn't know, or we didn't know, what viruses were actually injecting. Were they injecting protein or were they injecting nucleic acid, either DNA or RNA? That wasn't known. But they had to have been injecting one of the two because they're only made of protein and nucleic acid. All right. So this leads us then to the Hershey-Chase experiment. So in 1952, Alfred Hershey and Martha Chase showed that DNA is the genetic material of a phage known as T2. So T2 is just a bacteriophage that specializes on infecting E. coli bacteria and various other types of bacteria. All right. So what Hershey and Chase did is they designed an experiment that actually showed that only one of the two components of T2 was being injected into the bacterial cell. Now remember, a phage like T2 is only made of protein and nucleic acid. Hershey and Chase designed an experiment to show which of those two that virus was injecting into the E. coli cells. All right. From their results, they concluded that the virus was actually injecting DNA. And as a result of that, they concluded that at least in the case of phage T2, DNA was the genetic material because whatever the virus was injecting into the bacterium that was reprogramming the bacteria had to be genetic material, or else it wouldn't have been able to reprogram the bacterium. All right, so this is Hershey and Chase. This is the video that does not work, but again, this video is up on Blackboard, so you can go there and watch it. All right, so what I'm going to do now is explain their experiment. Okay, this is their experimental setup. So this can be a little bit complicated and a little bit confusing, but the important thing to remember here as you're trying to understand this experiment is what they were trying to do. They were trying to figure out or identify what viral component, protein or DNA, was actually being injected in the bacterial cells. The way they did that was by labeling the protein and the DNA differently. If you label something, maybe you can see where it goes, even a microscopic molecule. All right. How did they label these molecules, the DNA and the protein? They made them radioactive. And if you make something radioactive, you can track it with a machine like a Geiger counter, for example, that detects the radioactivity coming off of that substance. All right. Okay. So let me go through this. They basically did 2 runs or 2 batches of this experiment. All right, so let's look at batch number 1, which is the top one here. Okay, in batch number 1, they used radioactive sulfur 35. So sulfur 35 is an isotope of normal sulfur. We talked earlier in the class about how isotopes are typically radioactive. All right. Now, protein, if you remember back from chapter 5, has sulfur in it. DNA does not have sulfur. So in batch number 1, they grew viruses, T2 phage, in a culture medium containing radioactive sulfur 35. So when those viruses reproduced, they needed to make more viruses. They needed to build the protein shells, and so they incorporated the only sulfur present to make their protein, sulfur 35, which made their protein shells radioactive. But because the DNA inside the phage head does not contain sulfur, when the DNA was reproduced or replicated to make more viruses, the DNA did not become radioactive. So in batch 1, they've got radioactive protein coats. All right. Then, once they had mixed the bacteria in with the viruses, they allowed enough time for infection, maybe a half hour or so. Then they put the whole mixture in a household blender and they turned it on. The point of that was to separate the protein coats from the bacteria once the bacteria were infected. All right. Then they put the whole thing in a centrifuge and they spun it at high speed. And because the bacteria are heavier, more massive, denser than the viral coats, the protein coats, that caused the bacteria to collect in a pellet in the bottom, and the viral protein coats stayed in the fluid on top. Now they had the protein coats and the bacteria, and whatever was now in the bacteria from the viruses, they had the two separated. All they had to do then was look to see which of the two was radioactive. What they found is that the fluid above, called the supernatant, where the phage ghosts, okay, the empty protein coats of the viruses were, they found radioactivity there. They did not find radioactivity in the pellet, and that's a key point. If there's no radioactivity in the pellet, that tells them that the radioactive protein did not go into the bacteria. So all that first batch told them then is that the protein from the viruses did not go into the bacteria. It didn't tell them what did. They needed a second run or a second batch of the experiment to show that it was DNA that went in. So here, they did it again, but this time they used radioactive phosphorus 32. Why? Because DNA has phosphorus, protein does not. So when the viruses grew and reproduced, they needed to build more DNA, and so they incorporated phosphorus 32 into their DNA, and the DNA inside the phage head now became radioactive. The protein coats in this case are not radioactive. They allowed enough time for infection, put it in a blender, separate the protein phage ghosts from the bacterial cells, centrifuged it again, and looked for radioactivity. This time they found it in the pellet. And that tells them that the DNA, which is radioactive, must have gone into the bacterial cells. All right? Proving, essentially, that at least in the case of T2 virus, the T2 virus was infecting the bacteria with its own DNA. In addition, when these viruses that were produced inside these infected cells went on to reproduce and infect more cells, they always found it was the DNA that was radioactive and never the protein. So this experiment basically proved beyond a reasonable doubt that at least in the case of this virus and this bacterium, DNA was the hereditary material. All right. Again, that's a little bit of a confusing experiment, but you have to realize, to remember, that they needed to label protein and DNA differently to see where they went, and that's why they made them radioactive. Sulfur 35 in protein, radioactive phosphorus 32 in DNA. Again, the video for this, which is very good, is on Blackboard. You should go watch it. All right. So now we're going to talk about the structure of the DNA molecule. Okay. So we hit on this a little bit, back in, I think it was chapter 5, where we talked about macromolecules. But the structure of the DNA molecule is that it's a polymer. All right. Again, a polymer is a long molecule made up of monomers, smaller subunits linked together. Okay. The monomers in DNA are called nucleotides. You see the nucleotide right there. Every nucleotide is made up of 3 components. We have what's called a nitrogenous base. It can be one of 4. In this case, you're seeing guanine. It could also be cytosine, adenine, thymine, but in this nucleotide, it's guanine. So we have a base, one of 4. We have a sugar. In the case of DNA, it's deoxyribose, and there it is, 5-carbon sugar. And we have a phosphate group. All right, so base, sugar, phosphate, all covalently bonded together. That's a nucleotide. All right, now this nucleotide is linked to the next nucleotide here by a bond between the phosphate of this nucleotide and the sugar of that nucleotide. This nucleotide, again, linked to the next one by a bond between the phosphate and the sugar. That bond is called a phosphodiester bond, all right? Phosphodiester bond. Okay, it's a covalent bond, but it's always between the phosphate of one nucleotide and the sugar of the next. So what we have then is a structural component of the molecule called the sugar phosphate backbone, all right? The bases come off to the side as a side chain, and it's the sequence of bases, for example, guanine, cytosine, adenine, thymine, so G, C, A, T in this case, that makes up the genetic code. That sequence of bases is a language, and we know how to read that language. That language codes for amino acid sequence of proteins. Remember, amino acids are the monomer building blocks of proteins, right? Okay. And that's something that we would talk about in chapter 17, although we're not going to get to chapter 17. Okay. So, next thing to talk about on this figure here. Notice that it says that one end of this strand is the 3' end, and the other end is the 5' end. Okay. This has to do with the fact that there's directionality in the DNA molecule. The information in DNA flows in one direction. In the same way that in English language you read from left to right on a page, information in DNA flows from 5' direction, or 5' end to 3' end in that direction. So what do these 3' and 5' refer to? The carbons in the sugar. That's carbon 1, there's 2, there's 3, 4, and then there's 5. So for this sugar, this is the 3' side, that's the 5' side. For that nucleotide, this is the 3' side, that's 5' side. For this strand, this is 3' in because 3' carbons furthest one down. That's 5' in because 5' carbons furthest one up. It's just like saying this is the right hand end, this is the left hand end. That's all it is. Don't make it more complicated than that. All right. Now, what you're seeing here is just a single strand of DNA, okay? Normally, in terms of the way that DNA is found inside of our nuclei of our cells, there's another strand over here, all right? Another strand over here, and the bases are bonded together in the middle with another sugar phosphate backbone on this side, okay? All right. We'll see more about that here in just a little bit, all right? Okay. Now, let's go back to some of the research into the structure of DNA. All right. Prior to Watson and Crick doing their work, there was another researcher named Erwin Chargaff. Let me write his last name down here. Chargaff, okay, Erwin Chargaff. Chargaff and his research team were the first to look at base composition of DNA. In other words, to look at a strand of DNA and see which of these bases it's made up of and how much of each there is. His group was the first to determine that there are actually differences in base composition between different species, okay? And that actually, made DNA a credible candidate for the hereditary material, because maybe these differences in base sequence composition might be responsible for the differences that we physically see between species. And it turns out that's true. All right. What Chargaff also found is that in any species, the amount of adenine in a strand of DNA always matched the amount of thymine, and the amount of cytosine always matched the amount of guanine. This suggested a relationship between adenine and thymine, and a relationship between cytosine and guanine. All right. So these are what are referred to as Chargaff's rules. The amount of A matches the amount of T, and the amount of C matches the amount of G. Now, the reason for that, why that relationship is there, is not understood until the discovery of the double helix structure by Watson and Crick. All right. Okay. So after DNA was basically accepted as the genetic material through experiments such as what what Hershey and Chase did, after that, the challenge was to determine how the structure of the molecule actually accounts for its role in heredity. Well, here's another research group we need to talk about, a really important research group. Maurice Wilkins was the head of a lab at King's College in Cambridge in the UK, Cambridge University. And Rosalind Franklin was a relatively young researcher working in his lab. Franklin was using what then, late 1940s, early 1950s, was a brand new technique called X-ray crystallography. and she was using X-ray crystallography to study the structure of various molecules, but including DNA. So how does X-ray crystallography work? Well, essentially, the idea is that you have a molecule, say like a DNA molecule here, and you pass a beam of X-rays through a sample of that molecule. It could be in crystal form, it could be dissolved in solution, whatever. But you have a sample of the molecule, you pass X-rays through it, and on the back side, there's a photographic plate. So a plate with photographic film on it. Well, X-rays are a form of light energy, and when they hit the molecule, the X-ray beam is deflected. Okay, the atoms in the molecule cause it to deflect, and it literally draws a picture of the molecule on the X-ray film, or on the photographic film. This is called X-ray diffraction, okay, or X-ray crystallography. And Franklin was the first, I believe, or one of the first to actually use it to look at DNA and the structure of DNA. All right, so doing this, Franklin developed various pictures, a lot of pictures actually, hundreds of the DNA molecule. All right, so this is Franklin here on the left, Rosalind Franklin, about the time she was doing her work. And this is one of her most famous pictures of the DNA molecule. Now, I've looked at this picture a lot of times over the years, and while I'm no X-ray diffraction expert, here's what I think you're looking at. Again, imagine the DNA molecule as a spiral staircase, right? Imagine you lie on your back on the floor and you slide up underneath the staircase, one of those metal staircases, and you look up through it. What would you see? A circular or cylindrical structure with these steps coming in and sort of spiraling up inside of it. What I think you're seeing here is one sugar phosphate backbone, the second sugar phosphate backbone, and these are the bases paired up in the middle, like the spiral staircase, okay, like the steps of the staircase. All right, so from that photograph and various others, Franklin, who was super, super smart, I mean, really intelligent, she was able to figure out a lot of things about that molecule, certain measurements, the spacing of nucleotides, all kinds of things from that. Now, Maurice Wilkins, as I said, was the head of the lab that she was working in. Watson and Crick knew Maurice Wilkins. They knew that Wilkins and his group were working on the structure of DNA just like they were. They also knew that Franklin was using a new technique, X-ray diffraction, to look at DNA structure. They didn't have the ability to do that in their lab. So they paid Wilkins' lab a visit one day just to, say, hey, how you doing? Talk shop, that kind of thing. And I believe it was Watson who had an eidetic memory, what you might call a photographic memory. And Franklin, during that visit, showed them various pictures, like I've shown you here, of the DNA molecule that she had taken. She didn't give them copies to take back with them to their lab. She just showed them to them. Well, Watson used his eidetic memory to sort of take a visual or mental snapshot of her photos. They go back to their lab and use her photos to fill in the missing pieces or the gaps that they had in their molecule or their model of the DNA molecule. Now, that's all fine and everything, except when researchers get information from other researchers like that, they're supposed to give them credit for it. Franklin did not give them permission to use her data in their research. They did it anyway. They used her information without her knowledge to fill in the gaps in their study or in their research, basically finalized their molecule or their model of the DNA molecule and wrote that paper. Now, because she contributed so much, although she didn't know she did, she should have been at least a second author on that paper. Maybe Watson would be the first author. She should have been second, then Crick maybe third. They didn't give her an authorship, which is a big deal. Instead, they gave her, along with various other people, a simple one-line acknowledgement at the very end of the paper. That's all they gave her. They didn't really give her credit for it. They stole her work and didn't give her credit for it. They go on about 7 or 8 years later, and based on this research, win the Nobel Prize, which today comes with a multi-million-dollar award for each scientist involved. She didn't get credit for it. She missed out on that. What did she get? She died, I want to say, in 1959 or late 50s of ovarian cancer, and they think it was linked to the chemicals she was doing, or using rather, in her research. So that's just a complete travesty of justice, and it's one of the seedier sort of episodes in the history of biological research in the 20th century. Watson and Crick got a lot of crap for that over the decades, and Watson, who's still alive, rightly so, does still get crap about it because he was such a jerk about that. It nearly ruined his career, and Crick's as well, which it should have. Okay, I'll get off my soapbox now. So anyway, Watson and Crick put together their model based on Franklin's work. Franklin had contributed a lot as well, obviously, to this. Basically, the X-ray images of Franklin's enabled Watson to figure out the width of the helix and the spacing of the bases on their own using her information, right? This was information that they didn't have before. The pattern in the photo suggested that the DNA molecule is made up of two strands forming what's called a double helix. And that's what you see here. So here's the more familiar double helix or spiral staircase structure of the molecule. We have one sugar phosphate backbone shown as a ribbon in blue. We have a second one here. And then we have the bases pairing up in the middle. And notice how that A always pairs with T and C always pairs with G. Those are Chargaff's rules, right? We talked about that. The fact that A pairs with T and C pairs with G explains why Chargaff found that the amount of A always matches T and the amount of C always matches G. That relationship is called base pair complementarity. All right, base pair complementarity. All right, and you see it right here. This is the DNA molecule unwound, laid out flat like a ladder, for example. We have one sugar phosphate backbone here. We have a second on the other side. They run parallel to each other, and we have the bases paired up in the middle. A with T, C with G in all cases. All right. Also notice that if this is the 3' end, or that's 3' carbon there, of this strand, and that's the 5' end, that's 5' carbon here, on the other side, it's upside down. 3' end is on the top, 5' end is on the bottom. That is a situation referred to as anti-parallel. The two strands of DNA are anti-parallel to one another. They run in opposite directions, okay? That has really important implications for the way that DNA copies itself, which we'll see in the second video for this chapter. All right. Also notice that the hydrogen bonds between A and T, there are only two of them, right? Always just two of them. There are 3 hydrogen bonds between G and C. And that's important. That helps to dictate that only A and T pair up and only C and G pair up, as we'll see more about here in a little bit. This is a space-filling model showing you the various atoms in the molecule. It's giving you a sort of three-dimensional view of what it would look like if you could actually see it. All right. So Watson and Crick built models of the double helix to conform to the X-ray diffraction photos that they got from Franklin. And Franklin had concluded that there were 2 outer sugar phosphate backbones with the bases pairing together in the middle. So she already had a lot of this worked out, like I said. Watson built a model in which the backbones were anti-parallel. Their subunits run in opposite directions. So that was a unique contribution on Watson's part, but he probably wouldn't have come to that conclusion without having looked at Franklin's X-ray diffraction photos. Now, at first, Watson and Crick thought that maybe adenine pairs with adenine. So if you have an adenine here, maybe you have an adenine there, right? But those pairings did not result in a uniform width of the DNA molecule. So notice that adenine is a double ring structure. So is guanine. Cytosine is a single ring, so is thymine. So you always have a double ring with a single ring, double with single, and that keeps the width of the molecule the same at every point along its length. If you had adenine with adenine, or adenine with guanine, that would cause a bulge in that part of the strand because the 2 nucleotides would be wider at that point. If you had a thymine with cytosine, both single rings, that would cause sort of an indentation in the molecule at that point because those nucleotides are narrower. Well, Watson and Crick nor Franklin didn't see any evidence of the molecule being various widths along its length. It was the same width everywhere along the length that they measured. And that suggested, again, that maybe A pairs with T and G pairs with C. All right, which again conforms with Chargaff's data where he said the amount of A matches T and the amount of C matches G. See how they took various lines of evidence there to basically build a picture of what must be going on. All right, and here's that idea I was talking about in terms of the widths. So in the case of having, say, an adenine and a guanine, which, by the way, are the double ring structures, are purines, okay, you have the molecule being very wide there. In the case of two single rings, which, by the way, are called pyrimidines, you'd have a sort of pinched-in area here, right? It's only where you have a purine with a pyrimidine that you have a constant width along the length of the molecule, which again conformed to the X-ray diffraction data that Franklin had collected. All right, so Watson and Crick then reasoned that the pairing was more specific, again dictated by the base structures, adenine, thymine, cytosine, guanine. They determined that the adenine paired only with thymine, guanine paired only with cytosine, right? And again, that conforms to Chargaff's rules, where the amount of A matches T, the amount of C matches G. Because if you have one A on the left, you have one T on the right, there's a one-to-one relationship, okay? All right. And that's what we see right here. Okay, so I mentioned before that one of the reasons why adenine and thymine pair up is because they form two bonds. The structure of these two bases means that they both tend to form two hydrogen bonds, so they're compatible with each other. They each like to form the same number of bonds. Same is true with guanine and cytosine, except their structure generally dictates that they form three hydrogen bonds. They're compatible in that sense. If an adenine tried to bind to a cytosine, the difference in bond capacity would make that association unstable and they wouldn't stick together, right? So this base pair complementarity, which is based on their bonding compatibility, 2 here versus 3 here, is the whole basis for how DNA stores information, as we'll see in the next half of this chapter. But that is based simply on their structures, the fact that these two like to form 3 bonds and these two like to form 2 bonds. If that weren't the case, the DNA molecule probably would not be good at storing information. There'd be no coding system in it as a result. All right, so I think that's a good place to stop for this first half of this video lecture. We'll pick up with the second half talking about how DNA actually copies itself.